So I'd like to introduce your first speaker. It's Dr. Michael Steele. Many of you may remember Dr. Steele from his time here in Alberta. Dr. Michael Steele is an associate professor at the University of Guelph and holds an NSERC Industrial Research Chair in Dairy Cattle Nutrition. He completed his PhD at the University of Guelph and worked for Nutreco Canada Ag Research for 2 years prior to returning to academia. His current research focuses on the mechanisms that control gastrointestinal health and development in cattle, and he has published over 90 manuscripts in his career. So we are very excited to bring him here for you today, and I will let him take over now. Good morning, Mike.
Good morning. Thank you very much for the introduction. It's a pleasure to speak here virtually to everyone. I'm glad to see so many people live online. I look forward to having many questions. So the focus of my presentation today is first to showcase why I think it's really important to pay close attention in early life nutrition. So I think there's a lot of opportunities to improve on your dairy operation there. I'd also like to discuss some of the newest research that we've been doing and publishing in the field of colostrum nutrition, followed by milk feeding, and then weaning, and then talk about something that I don't think many people have really shared a lot of this research on, looking at alternatives to antimicrobials. But first things first, let's start with why I think early life nutrition is really critical in the dairy industry. Now, the concept of early life nutritional programming is not a new concept in other species. We've known that through nutritional stimuli in early life, this can have long-term consequences, okay? Now, just to give you an example of how this works, I like to think of, an animal as it's conceived as a ball rolling down a hill. And depending on different nutritional stimuli, this can have a profound effect for the entire lifetime of that animal. And just so things are closer to home, I'll just give you a good example. This is a story of some Dutch twins, let's just say, for the lack of a better word, the Schreiber twins. They were actually separated at birth, and one was fed 4 liters of colostrum at birth, and this affected it, and this is actually why it's this ball of this one brother has left on the left side of this valley here. And this really, through this this early life intervention, has created a pretty good body condition score, really good temperament, high IQ for sure, but a behavioral aspect is always in the barn. Now, the other brother was only fed 2 liters of colostrum, and that definitely affected the path of that brother long term. And as you know, the body condition score is a little bit higher, still reasonably healthy. Poor temperament though, average IQ, and kind of always in meetings and always wearing these jackets now. So I hope that this kind of, helps you illustrate the differences of how an early life program in nutrition can have a long-term consequence. And we see this also in the dairy industry. And I think the most closest relationship would be average daily gain in the pre-weaning period is associated with lifetime production. Now, there's many studies showing this. There's some studies that actually show no difference, but there's yet to be one study that shows any negative consequences of improving growth in early life. Another issue in the calf industry, before we get to this, I just want to poll and set up a poll with everyone right now and see how many people are actually are weighing their calves out there. So we're just going to go to a quick poll right now that I'm going to launch. So I want to know, do you weigh your calves and calculate average daily gain in the pre-weaning period? Yeah, so we're getting tons of responses here. So great. So this is actually pretty impressive, okay? I'm just going to stop the polling right now. But yeah, it's really impressive here in Alberta. I think that there's a lot more producers actually, weighing their calves compared to everywhere else around the industry. So kudos to everyone in Alberta for doing such a great job in that aspect. What you really do notice around the world, though, is that there's not a lot of average daily gain measurements occurring. Another thing that you also notice is that there's lots of gut health issues. There's still high mortality and morbidity across the board in a lot of the surveys during the pre-weaning period. The immune status, a recent study even from Canada, showed that 24% of calves are failing passive transfer, and that was published this year. And also antimicrobial use is still at an alarming rate, and we see way too much of this in calf nurseries. we see way too many of these images of overusing antimicrobials, and I'll talk about that later on in my presentation. So I had the absolute pleasure of working at the University of Alberta in starting my NSERC Industrial Research Chair with Alberta Milk. And we were able to, study colostrum management, planar nutrition, milk and solid feed intakes, looking at alternatives to antimicrobials, and also looking at the impact of the dam on the calf. So it was a great fun. Alberta Milk was the best partner I've ever worked with, to be very frank with everyone, still is really fantastic. And I also partnered with Lululemon Animal Nutrition to contribute more funding to our NSERC Industrial Research Chair that we had together. And I was really, really happy that all the other Western milk boards joined in on this project. And I was in addition to WestGen. So Sask Milk, BCDA, Dairy Farmers of Manitoba, thank you very much for supporting this project. And I hope you're happy with the fruits of the labor. Also, Alberta Agriculture has funded a lot of this research in addition to the NSERC Industrial Research Chair. So special thanks to them. And I was able to move it to the University of Guelph. And I thought it was important for Alberta Milk to start another IRC with Anna Larman, the University of Alberta. So I got funding from Elanco and Trow Nutrition to bring this chair to the University of Guelph to continue. So a lot of the work that we've really focused on is in colostrum nutrition. Now, we've done lots of work looking at different quantities. We're beginning to look more at quality a lot more and the potential for enrichment of colostrum. The quickness, we've done a lot of experiments. Actually, the first colostrum experiment we did at Brevely, the Deguis. Thank you very much. We did tube versus bottle feeding. I'm sure you've seen some of those results. And we've been really looking at cleanliness, and I've been collaborating with Lei Lu Guan looking at pasteurization and the impact of pasteurization on colostrum. So lots of work, and if you want a full collection of this work, I'd be happy to share it with you. I just want to share some of the most recent work that we've been doing though. So I think it's great that many farmers are doing Brix refractometers on farm, and I think it's important not just to do the colostrum, but also the calf and make sure that they can pass, they don't have a failure of passive transfer. This is some work that we just published. It just came out of the Journal of Dairy Science, where we looked at Brix refractometer readings in the blood of calves in the first week of life. Calves that were fed maternal colostrum versus colostrum replacer, because we kept on saying or hearing from the field that, okay, if you feed colostrum replacer, your Brix values are really low, so they're failing. So I think it's great that we're using Brix, but we need to use Brix properly. And as you can see from these two graphs on the maternal line, that's the only, I would only use a Brix if you're feeding maternal colostrum. If you're not feeding colostrum, maternal colostrum, you're feeding a replacer of some sort, I don't think it's accurate based on this graph on the right to really use a Brix refractometer with an R-squared of 0.4. And also what you notice is that although the serum total protein is low, the immunoglobulins are higher per gram of deciliter of protein. So it's really not fair to look at, using a Brix to assess if you're using a colostrum replacer. You do have to adapt your thresholds, but in general, an R-squared of 0.4 is not acceptable. So I would be sending away some samples to look at pure IgG if you're using a colostrum replacer if you want to be assessed. So this is just something that we wanted to point out. We noticed a lot of people were disappointed or even getting fired in the US because of low total proteins, but take home message, if you're using colostrum replacer, look at serum IgG. Now, we're just looking at IgG. There's so many other proteins in colostrum. And we're just beginning to realize what they do. We're doing a lot of work right now on insulin in colostrum and trying to determine why is it so high in colostrum? It must be sending a signal to the calf for some reason. We're trying to figure that out right now. We're also looking, and with collaboration from Mike Dugan's lab at Lacombe, Alberta, looking at how colostrum changes to whole milk with respect to fatty acid composition, which changes a lot, which is very exciting, because we're doing a lot of work in this area in the milk replacer field. In addition to the fats changing, and again, the point I want to make is colostrum is a lot more than just immunoglobulins. It's these proteins, it's all these fats. There's also a lot of carbohydrates that are not lactose. So instead of lactose, a portion of the lactose is replaced with these oligosaccharides, which are prebiotics. So this is just characterization study that we did at Brevely Farms, just collecting the first, second, third, fourth, fifth, milking from cows and looking at concentrations of these carbohydrates or being oligosaccharides, which are the prebiotics that you would see in colostrum. And as you can see, even in the first, second, and third milking, they're elevated, and there's differences in parity, which we're trying to uncover more so what could be regulating the total amounts. But it made me think after seeing that second, third, and sometimes the fourth milking with a lot of these bioactives that we should really be feeding not just pure colostrum and then switching to a whole milk or a milk replacer. We really need to transition because that's what the cow naturally does. If you look at a lot of these graphs, or if you look at insulin or IGF-1, it's still high in the second, third, fourth, fifth milking. So we shouldn't be discarding this. We should be feeding it to this animal. I think it's really important to supplement. So we did the study at the DRTC. This is where you go to do elegant work. Lots of great research has definitely come out of the DRTC. And in this experiment, we had all the calves fed the same colostrum in the first meal of life. Then we switched one group to a whole milk that was pasteurized in the second meal of life. And then another group to 50% milk, 50% colostrum. So kind of mimicking a transition milk. It's a little bit exaggerated though, but we're trying to mimic it there. Then also colostrum. And then we dissected these calves at day 3 of life. As you can see, the abrupt transition from colostrum to a whole milk depressed villi development. So these little finger-like projections in the intestine where all the nutrients are being absorbed. So I think that this depression is actually a really negative thing in a calf because most calves are really not growing very well in the first weeks of life. This could potentially be increasing the amount of nutrient absorption if we improve the intestinal development. On a more practical note, in the same experiment, we also looked at immunoglobulin levels in blood. So they were all fed the same meal in the first hours of life. So that's why the immunoglobulin levels are the same in all these treatments. But we fed a very small meal at 12 hours. So almost half the size of that first meal. And you can see, and to our surprise, there is still a considerable increase in immunoglobulins through feeding that second meal colostrum. So that take-home message here is definitely feed a second meal of colostrum at that 12-hour mark. And another important finding here is that these calves that were fed colostrum and all the way up to day 3 of life, they had a higher persistency of immunoglobulins in their blood in this particular experiment. So even though the gut may be closed at day 2 and day 3, having a source of immunoglobulins might actually decrease the amount that's trafficked to the gut in day 2 and 3. So it's something to consider. There might be some value to feeding some of this colostrum even after the gut is closed. And you're seeing a lot of this transition programs, even here in Alberta. I noticed a great video at the Vermeer farm. I saw Jake had a great video just showing that his program with this transition and his content with this program. So I think it's something to definitely check out and consider on your farm. I'd like to switch now to milk feeding. Now, I try not to talk too much about level of milk feeding, and because I feel like we've discussed this a lot over the last 5 years together. I think you know where I stand with respect to feeding, and I'm really happy with all the work that we did at the University of Alberta. And this is the new facility that was put together 5 years ago, largely due to Jen Hason's work, not mine. And also with Devakar Ambrose and Masahito Oba. I was really happy with so many experiments that we could do with all this automation. And this is just the one finding that I want to highlight, because I've highlighted a lot of our studies looking at the regulation of high milk intake, but it's really hard for me not to prescribe feeding more milk in the first month of life. And this graph is the only reason why. So this was calves fed through the calf rail at the DRTC, fed either 10 liters or 5 liters, okay? Now these calves even fed 5 liters, they grew incredibly well because their starter intake was really exceptional at week 4, 5, 6, 7. But week 1, 2, 3, and even 4, they don't consume enough starter. And calves, I don't know a calf in this world can consume enough starter in the first weeks of life to really compensate for this lack of growth that a calf would receive if they're receiving more milk. Okay, so if you want more growth in your animals, especially in the first month of life, you have to feed milk. There's no other way around it. I think that's all I'm going to debate today. I think we've gone through this debate a lot, especially with Gert Shriver. But this is why I truly believe that it's important to feed more milk, especially in this first months of life. Now, I've done a lot of research and we've looked at feeding larger meals a lot, and even feeding 2 times a day. If you don't have automation, you can still feed these meals of 4 liters without having insulin sensitivity. And that's some work that my first MSc student at the University of Alberta, Jane McPherson, published. But sometimes just like practical considerations on farm, and I think the advancement from a 2 to a 3 liter bottle to now to a 4 liter bottle is just probably even more significant than some of the work that we've done through this IRC. But I think we really need to get away from feeding this misconception that calves can't be consuming these large meals. So even in 2 time a day feeding, they can consume 4 liters per meal, even in the first weeks of life when they're not consuming starter. So just because you don't have automation doesn't mean you can feed more milk in the first weeks of life. I think it was really timely, especially with the forecasts. Even though I live in Ontario now, I still follow what's going on in Alberta, and I know what's hitting you right now with the -35 weather that's coming your way. I think that this is an important part that I don't really put in a lot of my presentations, but I just wanted to touch base on. You really need to adjust the feeding rates during the winter. Okay, so you can do that by increasing the feeding frequency by adding a meal, or you could be increasing your total volume per meal, but it's really essential. And this is a chart that I got from Dr. Mike Van Amberg from Cornell University, just showing the amounts of milk replacer or milk dry matter required to meet maintenance requirements in calves during cold temperatures. So let's just look at this 55 kilogram calf, 0.5 kilograms of milk replacer required just to meet maintenance at 20 degrees Celsius. It requires double that this weekend, everyone. So you have to adjust your milk feeding rates during the winter everywhere, especially if they're outside in hutches or even in a cold environment that's inside. You have to adjust this. You know, coats work fine, but I would go here first. I think it's deep bedding and making sure that the milk is delivered. And if you put a coat on top of that, that's wonderful. But you definitely need to be feeding them more milk during the winter. So that's all I'm going to say about feeding more milk. I try not to get too carried away with that topic. I have a tendency to do that. But this is another exciting topic related to milk nutrition in calves for 50% of the farmers around Canada and that are feeding milk replacer. So when I look at milk replacer formulations, and this is just a general overview, not all milk replacer companies have these types of formulations, but in general, the lactose is always higher and the fat is always lower when you compare it to whole milk. So the reason why is because if you feed more lactose and more protein to animals, they're going to grow more. But we were just wondering like what that actually would do to the gut. And our concern here was the osmoles within the milk replacer. We think that they're quite high, and we're concerned that the high lactose could be contributing to an osmotic pull causing a diarrhea in a calf. So we had those questions, and this was kind of reiterated by some work of one of my PhDs showing hypertonic milk replacer increases gut permeability. So this is Juliette Wilms, who just recently joined our lab. And this is some work that she did with Trow Nutrition. What the work that was done at the DRTC by Annemiek Welberin is that higher lactose results in increased gastric emptying and lower glucose tolerance in the first week of life. So if you're feeding 2 times a day, you're feeding these larger meals in the first week of life, if you have a high lactose milk replacer, it could be reducing glucose tolerance. So this was an interesting finding. The long-term consequences of this, we don't know. This is only the first week of life, but it's more of a proof of concept. So I want everyone to be very cautious interpreting these results. They're not saying the current milk replacer formulations are wrong, okay? They're just saying perhaps there's reason to study it a little bit more. So that's what we've been doing more and more of. And we've been, questioning, those are all restricted feeding regimens that I showed in the previous slide. What would happen in an ad libitum setting? So we did an experiment at Trow Nutrition in the Netherlands, just outside of Boxmeer. And what we did is we wanted to see if an ad libitum intake in these calves were consuming enormous amounts of milk replacer through an automation system. So if you're over 1 kilogram a day, that's really high, but some of these calves are actually consuming up to 1.6 kilograms on a dry matter basis. What we found here is that actually the high fat calves consumed less milk replacer when offered ad lib compared to the high lactose. And we're trying to figure out what regulates that, but it's most likely when you plot metabolizable energy, since the high fat is actually higher in energy, this is probably how the calf can actually regulate its own intake. It's actually just trying to meet its metabolizable energy intake. So what's compensated with respect to energy density and the higher fat being higher, you're noticing that these calves actually consume a little bit lower. And that's what you see in typical ad lib. There's nothing wrong with it, but just an interesting phenomena done by my MSc student, Juanita, who's actually was raised in Red Deer. Some other work with milk replacers that we find very interesting and we've repeated in many experiments is that these high fat milk replacers actually increase gut permeability. Now, some people might think that that's negative. We don't really know if it's negative or positive at this point. We just know that a lot of the high fat milk replacers, when you're increasing the fat, you're going to cause a leakier gut. And how you measure that is dose an animal with a compound or chromium, and you look at how that chromium shows up in the blood. If it shows up faster and at higher concentrations, it means you have a leakier gut. So this is happening, which has led to a lot of research by our lab in this area that we're currently doing an experiment looking at fatty acid composition in milk replacers and how that relates to gut function, because we think it could be really important. Something to consider is that the omega-6 and omega-3 ratios are strikingly different, and a lot of the polyunsaturated fatty acids are very different in a milk replacer compared to a whole milk. And we're thinking that this could be impairing gut function in addition to metabolism of these animals. So this is the bulk of a lot of our work. No need to be alarmed with milk replacer formulations right now, but we just think that there might be some potential in the future to even improve them and even program animals differently. So let's move to our next section being on weaning. And this is a fun area where we've done a lot of work, and we focused a lot of our initial studies looking at ruminal development. The take-home messages from a lot of this work is that you should be weaning animals later in life, so after 8 weeks of life, if you're feeding more milk. You shouldn't be weaning them earlier. So we showed that in several experiments through the IRC, and also step-down programs. You definitely need a step-down program. Take-home message here is it should be longer than 2 weeks. And if you can have less abrupt transitions or less steps or more steps, so there is more of this gradual transition, it will result in a better weaning transition. So this is where our work is now. And it's all because if you wean these animals early and abruptly, and this is a perfect example of calves that were weaned really early in life at 6 weeks off high milk, and really abruptly just a 1 week step down, they're just not going to be able to consume enough feed to actually gain at a proper amount here. So this is actually work done by Jolette Van Nijkerk, who was my first PhD graduate from the NSERC IRC, and now she's an instructor at Lakeland College. So for the people, just while we're on this topic of Jolette, for the students at Lakeland right now, you better be listening up to Jolette, okay? You better be listening to her in her lectures because you can learn a lot from her, okay? But this is some of her work from her PhD that she defended this year. This is some other cool work that she did in the rumen of calves. So from the same calves, we actually put fistulas in them. We measured ruminal pH. What was really interesting about this, when you look at the time below different thresholds, this pH is really, really low. These are really acidic environments. They definitely have what would be termed in a mature animal ruminal acidosis during this time of weaning. But through time, they do adapt. Now, if you look at the rumen papillae in these calves, and this is papillae retracted from the calves, you'll see that there's not that much development even a week post-weaning, but through time, they eventually develop and they'll be absorbing a lot of these acids. So we've done a lot of work in this study just through characterization of what's going on in the rumen. So we're just in the midst of publishing some more work with rumen microbiology done with Lei Lu Guan. But we found this a very insightful study to look deeply at. But we also not only looked at the rumen, and this was the focus of Jolette's PhD, is really looking at the lower gut as well. And this was inspired actually by our next speaker, Greg Penner, where we did an experiment when I was working in industry where we showed that weaning actually induces a leaky gut. And this got me thinking that, you know, the structure of the rumen is actually quite strong even during these massive acid insults during weaning. You know, perhaps some of this lag in overall growth could be coming from intestinal damage and inflammation from that. And that all came from a study that I did before I joined the University of Alberta, looking at abrupt versus gradual step down weaning. And what was shown here is that animals that were weaned relatively early in life and not on a high starch diet, it was a 30% starch concentrate, they actually had really high fecal starch. So this makes me think that you're getting a lot of hindgut starch, which could be fermenting, and a lot of E. coli turnover. All these toxins are released, could be being absorbed because you've got a leaky gut during weaning. And then you have a whole animal inflammatory response. And this was really shown in a lot of the samples that we've been collecting during the weaning period. There's lots of variability in this, but you see a lot of this bubbly manure. And this really shows that you might have a lot of fecal starch in the manure, but also these calves probably aren't adapting very well to your transition program during weaning. So another issue, and I know I've talked about milk replacer composition, but also starch and the amount of energy in a lot of this, a lot of the starters that you're feeding are quite variable. So on the marketplace, they can range between 10 and 50% starch, when in a lactating cow, we're feeding around 28%. Now, this could be inducing ruminal acidosis and possibly a hindgut acidosis. So I think that we should really be looking at how starter composition is tailored to your milk feeding program, because I think there's a lot of interaction. And I think that we should be spending more time looking at the lower gut as well, and not just the rumen. So this is some of the initial work in this area done again by Yolette Van Nykirk, looking at high-low milk feeding levels and how they interact with the fermentability of the corn in the starter. So there is some mild interaction. In this study, I think these animals were weaned a little bit too early to really elicit the response based on our hypothesis that these animals fed whole corn off high milk would be damaged or have a lot of hindgut acidosis. But I think this is a step in the right direction, starting to look at this interaction. And also looking at the lower gut, which this is an image of Yolette when she's taking some endoscopic biopsies. You know, I think we're almost there. I'm gonna stop right now. And I noticed that there is a question from Courtney Felton. So I'm just gonna stop and get that question. Courtney, if you're still there. Okay, while we're waiting for Courtney, I'm going to actually launch another poll here. So I just have a question about forage intake, and Courtney, if you want to come online while we're launching this and we can talk, that's great. But I have a question about forage intake during weaning, so I'm going to launch that right now. So how much forage does a 100-kilogram calf consume at weaning? So just on average. Greater than 4 kilograms, greater than 2 kilograms, kilogram, less than half a kilo. So right at weaning. So about maybe like 2.5 months, how much forage are they consuming? Great, so answers coming in, tons of wow, we've got a lot of people on this call. I just noticed we've been increasing it quite a bit. This is great. Yeah, everyone, so we're at a lot of, I'm gonna end the polling. I'm gonna share the results right now. And what we're seeing is that a lot of people think about 2 kilograms of a forage is what a calf's consuming right around this weaning. Some people think over 4 kilograms, some think 1 kilogram. Not a lot of people think less than half a kilo right at the time of weaning. So I'm gonna go back, get out of this great polling feature. This is gonna make us really learn a lot about this. So just,
Mike,
yeah.
So there's nothing from Courtney yet, but you do have a few questions in your Q&A panel if you wanted to address those now.
Sorry, so should starters be higher in protein for high milk volume feeding? I don't think that there's really a lot of evidence for going over and feeding these high protein starters. And this was from JP and Martin Brower. When I'm saying high protein, I mean over 22%. I'm a little bit skeptical of those because I think that you might have a lot of ammonia in the rumen, and then the calf has to process all that ammonia, and that costs a lot of energy. So I'm not so convinced at that right now. However, I know a lot of people working in that area. So I wouldn't go out and feed these really 28% crude protein starters at this point, okay? So So could you also go higher with your grams per liter of milk replacer, is a question from Gideon as well. That's a great question, Gideon. I just don't feel very comfortable going over 150 grams per liter in a milk replacer. I think that's already quite concentrated. I know some people getting away with 160, but where it really falls apart is when people go above that level. So if you still have some wiggle room, that's one way of doing it. But I think an extra meal and increasing the meal volume, if you're not at 4 liters per meal, is another way of doing that. Garrett Harmon. Garrett, nice to hear a question from you. Does the fat content of the milk make a difference feeding higher solid content? Yeah, I'm not sure about the fat content of the milk feeding a higher. I think that I'm just going to speak broadly about milk fat at this point. And I think that we should be not underestimating the value of fat going into milk, whole milk, or milk replacers, because I think that the calf at that point really needs milk fat, and it's actually controlling a lot of metabolism at this time of life. And a lot of these thoughts I have is based on infant research and human research. So this is great. How many questions? I'm just going to charge through my presentation, this last part of my presentation, and then hopefully we have more time to do more Q&A. But as always, I love presenting in Alberta because there's no boundary when it comes to questions. Back to my poll question, a lot of people thought that calves can consume, sometimes even 4 kilos, but even 2 kilos of hay during weaning. You got to think about a 6 calf, that's actually not fed that much milk, has a rumen the size of a basketball. So only half a kilo, that will fill that entire basketball if you're feeding hay or a straw, and the calf won't be able to consume anymore, compared to, let's say, 5 times as much starter. You can see the space there. So calves actually consume a lot more concentrate at this time of life. You're not going to have a calf consuming over half a kilo right at weaning of any kind of forage, okay? That's really hasn't been recorded in a lot of literature. And if they are recording that, I would really be skeptical of it. I think you just need to consider that a calf at that stage is definitely feeding less than 10% of its total diet being forage. Okay, in the way that we feed them today. So you might think they might be eating a lot of hay or straw, but they're actually not. So that's why I think it's important to feed a lot of concentrate, especially during the weaning period. Now you can mix it, and this is 50% chopped straw and 85% concentrate mixture. It's a dry TMR that's become popular in Alberta, but it's not, you can do it with straw. You can also do it ad lib with straw and also concentrate. But I think they should be receiving these high concentrate diets. And this is some work done at the DRTC where we wanted to look at the interaction between pre-weaning feeding, so high-low milk again, 5 versus 10 liters of milk. And the classic response here is what you'd see is more growth with more milk, but you're going to have less starter intake because they're consuming more calories from milk. And the post-weaning diet that we took, half the calves fed the low milk and half the calves fed the high milk, we put them on in 70% concentrate diet, and also an 85% concentrate diet. So though it doesn't really look like an 85% concentrate diet on the right, it is, okay? So what we noted here is that with respect to dry matter intake, these calves really didn't have a lot of interaction between calorie intake pre-weaning and how they actually eat post-weaning, and actually how they grow as well and develop. But what was really intriguing is how much they could actually consume of these high concentrate, high energy diets post-weaning. Now, I'm not saying to feed them 15% forage for the entire period up to 25 weeks, but I'm thinking the growth rates that we were achieving in this area were over 1.5 kilos. Granted, a lot of that could be rumen fill, but they weren't having excessive body condition scores here in this first period. So I think feeding them these concentrate, these really high concentrate, the forage ratios in the months post-weaning, especially the month or two post-weaning, is really important to have really efficient growth, because their average daily gain is really high and their feed conversion is also very high at this stage of life. What was interesting is that this post-weaning diet actually had a lot more influence on physiological outcomes like IGF-1 and the onset of puberty and reproductive development, with some work that we published in the Journal of Dairy Science actually this year with Dr. Ambrose. But it was just made me think that we've been talking so much about feed restriction in the pre-weaning period, but typically we're feeding 2 kilograms of concentrate to these animals right here. And we're assuming that they're eating all this forage. So we're actually restricting energy intake post-weaning. The longer term implications of that, I'm not really sure, but it just is food for thought. So we really need to take this on and study things past weaning, because most of the research is the first 2 months of life, and we need to do more. Last but certainly not least on my list of things to go through is to introduce some of the newest research that we've been doing, looking at alternatives to antimicrobials. Now, this is a serious issue in the calf industry. The female calf industry, replacement industry, big issue. Male calf industry, also a very big issue. So I've been working with a lot of the male calf industry for a long time, actually, even before I was an academic, because I've been fascinated with these forgotten calves, the male calves, which are a huge part of our dairy industry, whether we like it or not. We need to really face this. And, you know, like they're all raised, they're coming from many different environments, and they all go through a natural challenge model. So I found this very interesting to study, because I want to alleviate these high fecal scores that you see in the first week of life in all veal industries or all dairy beef raising facilities. But what they typically do to compensate for this is they administer prophylactic antimicrobials. And in this case, this is a study that was recently accepted in the Journal of Dairy Science showing the use of neomycin. So we studied the short-term use of neomycin just for the first 14 days of arrival. Then we looked at a long-term response of the first 28 days. Then we had one group that didn't receive any. But we were still treating them. If they reached certain illness thresholds, we did treat them for animal welfare issues. But what you can see from this is that if you look at your defined daily dose for antimicrobial loads, this is a big difference with this prophylactic. The therapeutic antimicrobials, there's a little bit of a peak there at around 7 to 10 days in the ones that didn't receive any. But this is a big, big difference when you look at the amounts of antimicrobial actives that are going to these calves. And in this study, there is actually no difference in the incidence of diarrhea and other health outcomes. And also the therapeutic treatment of antimicrobials was also not different. The time for resolution was not different from diarrhea between all these treatment groups, but there was a mild difference in the amount of water in the manure when you're feeding the neomycin. The average daily gain was not different at all. The growth of the animals was not different between all these treatments. So we're going into the male calf industry, a lot of this, even with vet scripts now, we're still feeding a lot of antimicrobials. And I'd like to question, is it really necessary? So this is a lot of the work we're doing right now. We're also looking at alternatives, and one alternative we've been looking at is a yeast by the name of Saccharomyces cerevisiae boulardii, which can affect the microflora, the barrier function, and the immune function of the gut. And we showed through using this natural challenge model in a lot of dairy milk calf model that we've been using. This is at Grover Nutrition where we did the study, is that the fecal score, but also the total number of diarrhea cases was actually decreased through the use of this boulardii product. So that the boulardii is here in the green. Now we wanted to understand how this actually happens. And if you can't really do this in a commercial farm. So when you need to do some really technical work, where do you go? You go to the DRTC. And I luckily had an awesome postdoc. We did really intensive experiments from birth feeding these probiotics. And we found that it can influence a lot more things than just the microbial community, but also the host. One example of that is looking at a natural immunoglobulin produced by the gut of a calf. We call it IgA. Okay, so it's in colostrum, but in the calf, right at birth, doesn't produce a lot of it, but not that long into its life, it starts producing this IgA in its gut. And what we noted is that these calves fed the boulardii here in green actually had higher levels of IgA in the digesta of these calves. So this could be one mechanism why they have less diarrhea. We didn't really believe that. We wanted to look deeper, so we looked at the cells that actually produce the IgA, and they were also greater in the boulardii group. So this just goes to show it's nice to have these commercial settings, but when it comes down to it, you need to figure out the mode of action. And that's why it's wonderful to work at places like the DRTC. So this kind of concludes my presentation, just showing you some of the work in colostrum, plain nutrition, antimicrobials, which is growing a lot right now. And where we're heading in the next 5 years is looking at the maternal impact. And we've just set up some grants for the next 5 years, looking at, you know, what really affects, what can we do to the dam to affect the calf in early life. So we're really excited by that. Special thanks to my NSERC IRC team. They've went through a big transition for those who flowed from the University of Alberta to the University of Guelph. They've adjusted so well, and they're the reason for all of this hard work. It's not possible without them. And there's been a wide variety of them graduate and have gone on to do great things from this NSERC industrial research chair that we hold together. I'm really proud of them all. And also proud of our accomplishments. We're over 51 published work from this IRC when our goal was 25. So I was really happy with that. And we have over 100 presentations since 2016, just promoting the work that we've done together. And my best accomplishment, I think, there was a lot of dairy representatives from Alberta, was hosting a conference at the University of Guelph before COVID. So this is a pre-COVID photo, so you don't have to freak out. But yeah, this was great. We had great representation from Alberta Milk at this conference. And actually the most questions from all audience members were from Alberta Milk. So in review, I really want to thank everyone who supported my research in calf nutrition at the University of Alberta. I really want to thank my IRC chair mentors that have helped me immensely along the way. And these 5 people really helped me out when I started my career at the University of Alberta. So Ruud Zylstra, Leilu Guan, Masahiro Oba, Devakar Ambrose. And although not from the U of A, Herman Barkema played a huge role. So I'd be very happy to take any more questions if I haven't gone over time, and I apologize if I do, and I'll be sticking online after this meeting if there's any additional questions. So thank you very much.
Thanks, Mike. That was wonderful. We are going to cut you a little bit short on questions. We're a little over time, but I will offer, we do have some participants that are participating in phone only, so they can't enter their questions in the Q&A. For those of you participating via phone, to ask a question or raise your hand, just press *9 and we'll get a notification. So I'm just going to launch a poll while we're waiting for those questions to come in. And I will offer that for any of the questions that are in the Q&A currently, there's an opportunity for Mike to either answer those while the other presentations are going on just by typing a response in there, or we can leave them till the end. And if time allows, we'll have all our presenters back to answer questions live. And I'll lastly say, if we use all our time today and there's questions remaining from all the speakers, we have the opportunity to ask and answer these in our Ask an Expert feature, which is on the DRECA section of the Alberta Milk website. And to be clear, that's the public facing side of the Alberta Milk website. For those of you that are not familiar with our DRECA pages there. So yeah, we will make sure your questions are answered one way or another. And so yeah, got a lot of good responses coming in. Oh, sorry, and this is Greg's question, but hey, Greg, we're going to have some answers and I'll let you relaunch that one. I got ahead of myself. Sorry, folks. This is the question we want answered right now. So of course, for us, this is our first virtual DRECA event with COVID, and potential great opportunity here for us to do more of these in the future if this really resonates with our audience, knowing that our audience is producers, our farm service providers, and other partners. And if we're lucky enough, this audience extends beyond Alberta today with this virtual format. So I don't see any questions coming in from our phone call listeners right now, so we'll move along to our next presentation. Thank you to everyone for your responses in this poll. And thank you to Mike, and we look forward to having you here at the end of the session so that we can have some more Q&A with you. Awesome.
Sounds great, and I'll answer all the questions that are in the question and answer panel. Thank you.
Thank you very, very much. So I'm just going to do my introduction here for Greg, and Greg, you take over sharing your screen whenever you want. So we have Dr. Greg Penner joining us. He is a professor in the Department of Animal and Poultry Science at the University of Saskatchewan and holds a Centennial Enhancement Chair in Ruminant Nutritional Physiology. He received his bachelor's degree and master's of science from the University of Saskatchewan and his PhD from the University of Alberta. Dr. Penner's research focuses on forage utilization, beef and dairy cattle nutrition, and regulation of gastrointestinal function in ruminants. Individual projects range from a focus on fundamental aspects of physiology to those with applied outcomes that can help promote efficient and sustainable beef and dairy production. And I will already give you a heads up, Greg, that I have had 2 questions come in prior to the event for you.
Perfect. Thanks for the invitation. It's really exciting to be able to present. One, it's always exciting to hear Mike and to follow him. And certainly, Mike, I learned a lot from your presentation, and I look forward to some discussion after as well. So I hope I can carry that same type of enthusiasm as Mike presented. I'm going to talk today about transition cow nutrition, focusing mostly on automated milking systems, but realizing that not all producers are using automated systems. They're still, the minority of production. I'll focus on transition cows in general, and then we'll apply some of those concepts into the automated milking systems. Okay, so getting to the polling question, I think you already saw this. What I'd like to do is just hear your concerns and see your vote in terms of major concerns that you perceive with those fresh cows, and particularly that postpartum period. So do you perceive risk for ketosis being greatest risk, supporting rumen health, minimizing the energy balance gap, or managing those social interactions that impact nutrition? So maybe not even the nutrition per se, but actually addressing that social, environmental aspect that's related to those cows being able to consume the diet that is formulated for them. So I'll just give you a couple more seconds here for the poll. I think it's starting to slow down. Perfect. So let's share the results there. What I see is most producers are suggesting that that energy balance gap is really the major concern that they have. And I hope what I'm going to present addresses that issue. I think it does address that issue quite clearly. However, what I hope you also recognize is most of these terms were connected aspects. Obviously, if we can minimize that energy balance gap, we probably will reduce risk for ketosis. The only way to do that is through a program that actually supports rumen health. And obviously that's impacted by other factors, and stocking density would be one of the obvious ones that will impact the ability of cows to actually drive their dry matter intake and meet their energy demands. So with that in mind, we're going to continue on. And I really want to set the concept for where I'm coming from for this presentation. So we have 2 different programs that we see being used quite commonly in industry, either a 2-stage dry cow program where you may have a far off or a single and a close-up diet, or you may have a single dry cow diet. Now, a lot of these are being used. The reason I have single cow dry cow diets is in many cases, this is utilized in smaller farm scenarios, and it's also the example that I'm giving you in the diagram just to the right. We're seeing a lot of adoption of these Goldilocks, the low energy or controlled energy diets, particularly in the close-up phase. And then these cows are transitioning to either a fresh cow diet or directly onto the lactation diet. And so there's obviously some dietary challenges that occur at that time. So I just want to highlight a couple things with the diagram on the right. And this is from a recent publication from the Minor Institute in the Journal of Dairy Science, where they were revisiting these single dry cow diets. And so this is both a single diet for the far off and the close-up phase. And what I really want to highlight is this change in dry matter intake that is occurring for these cows as they approach parturition. So these are cows fed a high energy dry cow diet, so about 27% starch, pretty high starch for what we would typically see right now, at least for those controlled energies. And they compared that to cows that were either fed that controlled energy program, about 12% starch. And then half of the cows on that low energy program went to the high energy program for the last 3 weeks prior to parturition. So this is really that old concept of the steam up approach. Okay, I'm not going to talk about the outcomes of the response, but really what I want to emphasize is this reduction in dry matter intake. And this reduction in dry matter intake prior to parturition, I believe, is setting the stage for a lot of outcomes that we need to deal with. The other aspect I want to highlight is when you look at the response between those cows fed a high energy diet and those cows fed the low energy diet, those cows fed that low energy diet, while they have lower dry matter intake in this study, lower energy intake, they also have a more stable dry matter intake. And I think this stability in intake response is quite important. Okay, so why would we be concerned about the reduction in energy intake as parturition approaches? Well, I think there's a couple reasons. One, first of all, we're dealing with a change in energy intake. So that energy intake will be decreasing at a time when energy demand is still increasing. That initiates mobilization of body reserves, might increase risk for ketosis, and that low intake probably also increases risk for displaced abomasum, although there are many other factors involved in that as well. Okay, so this leads to polling question 2, and this really is getting at the other factors that might be implicated. And I think there's been a lot of work, and I'll show you some of the studies suggesting that transition dairy cows might actually be at risk for ruminal acidosis. And so I'm just curious to hear why those cows might be at risk. Could it be decreasing dry matter intake prepartum, a rapid increase in dietary energy postpartum, increasing dry matter intake, or not enough dietary buffer supply? I don't see big changes in the poll outcomes, so I'm going to end the poll here and share the results. And what we're seeing is a rapid increase in that dietary energy density postpartum being a primary risk factor. I think that's certainly one of them. But what I'm going to highlight over the next few slides is that reduction in dry matter intake prior to parturition probably is at least as great a risk factor as the dietary changes that occur postpartum. So hopefully I can convince you, or I look forward to the discussion if I haven't, to see if you do buy the concepts that I think our research is showing. Okay, so let's look at what happens in terms of risk for ruminal acidosis. So in this study, these are very low numbers of cattle in these studies. This is work I did as a master's student at the Lethbridge Research Station, where we were able to continuously measure ruminal pH in these transition dairy cows as they progress from the prepartum into the postpartum stage. This was really some of the first work where we had continuous pH measurement in transition dairy cows. And what we were able to show is we really have quite high prevalence rates. And our definition in this case was anytime pH is below 5.8 as our indicator for ruminal acid. So we saw, over or almost 80% of our heifers in this study actually experiencing ruminal acidosis within the first 21 days of lactation. So some pretty severe challenges potentially facing those cows. Now, when we overlay that same data, in this case, now we're not looking at prevalence, we're looking at the amount of time cows are spending below that threshold, and we overlay that with dry matter intake. So their dry matter intake response is shown in the blue columns. Their duration, that pH was below 5.8, is shown in the red columns. You can see that the response or the slope of those lines actually follow different patterns, where early on within that first 21 days, intake is still increasing, but we actually appear to reach a peak or a plateau in terms of risk for ruminal acidosis. And after that, it declines. Okay, so there's a number of reasons that this might be present, but we really do think that these early fresh cows are at quite high risk for rumen acidosis. we're not the only group that has looked at this. So this is data out of Austria looking at why cows would be at risk for low pH and early lactation. And there's a couple important factors that I want to highlight. In this case, they had no different dietary treatment across all the cows. So they were all fed the same close-up diet. They were all fed the same early lactation diet. They were simply trying to use a retrospective characterization to understand why some cows were tolerant and some cows were susceptible. One of the main factors that they observed was those cows that were tolerant actually had greater intake during the close-up period than those cows that were susceptible. The challenge to this is they did not observe any differences in postpartum dry matter intake or milk yield, questioning how important this difference in reticuloruminal pH really is. But I think the case has been made, or at least the suggestions have been made, that dry matter intake in the close-up period, and as I'm arguing, that change in dry matter intake as parturition approaches probably is the major risk factor. Okay, so why we came up with this concept is really some work that we conducted, in this case using beef cattle. And in this study, we were interested in understanding how short-term exposure to low feed intake would affect rumen function and regulation of pH and risk for acidosis. So in this study, we fed them voluntary intake, or we fed them 2 voluntary intakes, so ad libitum dry matter intake, and then we restricted them to 75, 50, or 25% of their voluntary feed intake. Now, you're probably already wondering, are these intake restrictions relevant for dairy cattle? Well, if you look at the literature, dairy cattle on average reduce their dry matter intake by about 30% during the last week of parturition. So that 75% treatment is probably pretty close, maybe even a little too low of a restriction relative to the average dairy cow. If you think of dairy cows that have other transition disorders like metritis, mastitis, ketosis, displaced abomasum, the 25% probably is not severe enough. Okay, so those cows really drop feed intake. There is nothing that is more potent at dropping feed intake than inflammation. Now, not surprising, when we drop feed intake, we see those cows drastically reduce the concentration of short chain fatty acids in the rumen. So there's less substrate going in the rumen for the microbes. And obviously, if there's less going in for the microbes, there's less to ferment. And so the outcome of that fermentation is diminished. Okay, so as fermentation is reduced, we see pH go up, and that makes a lot of sense. Okay, so as pH goes up more when cattle go off feed, and we can see that both in terms of mean pH and the amount of time that pH is below 5.5 in this case as an indicator for ruminal acidosis. So this is really showing that as those cattle go off feed, there's a reduction in that fermentative activity and pH goes up. The problem comes when those cattle go back onto feed. And so after that 5-day of exposure to low feed intake, we allowed those calves those cattle full access to their feed. We did not change the diet, and this diet only had 40% concentrate on a dry matter basis. You can see those animals restricted to the greatest extent during feed restriction were also eating less even when we allowed them to eat at a voluntary quantity. Now, despite that lower dry matter intake, we had very low pH in those cattle. And in fact, just allowing them access to their regular diet induced ruminal acidosis in all of the cattle that we had on this study. So you can see even those cattle restricted to 75% of their intake dropped mean pH to just under 6.2. You can see those animals that were restricted to 75% had a mean pH that was below 5.9. So at these levels, we would be assuming that we would start seeing negative effects on fiber digestibility, and we probably see some risk for the inflammatory response that we see with ruminal acidosis. Why we think this happens is because exposure to those off-feed events or those low feed intake events decrease the capability for those cows to absorb nutrients across the rumen. So this is the absorption of those short-chain fatty acids after being exposed to feed restriction. And we know that short-chain fatty acid absorption is not only critical for energy supply, but is also the primary mechanism that regulates rumen pH. So when we have feeding scenarios that decrease the ability of cows to absorb those short-chain fatty acids, we increase risk for rumen pH or for rumen pH depression. We can manipulate the response with diet. And so in this case, we were looking at the impact of feeding a high forage diet or a moderate forage diet after that period of feed restriction. So I'm just showing you the recovery response when they were fed ad libitum after being exposed to low feed intake. You can see those cows that were fed the high forage diet had a greater recovery rate for dry matter intake than those cows that were fed a moderate forage diet. And so we're bringing in some suggestions here that those transition cows, as they're moving from that prepartum phase and they drop intake into the postpartum phase, there may be an opportunity to accelerate the rate of increase for dry matter intake with short-term feeding of a high forage diet. Now, I think it's important that this is short-term because we certainly don't want prolonged feeding of a high forage diet as it would exacerbate that negative energy balance gap. On the other side, we can also look at the amount of time that pH was below 5.5, and it's very obvious. Not only did those cows that were fed the high forage diet during recovery have higher pH or higher dry matter intake, but they also had higher pH, and we actually nearly eliminated risk for ruminal acidosis in those cows. So bringing this back to the transition cows, obviously the model we used were non-lactating cows, non-pregnant cows, but I think the principles really apply here, and I think it justifies further work into the early fresh phase to see if there are strategies that we can use to help accelerate how fast those cows increase dry matter intake, and to ensure while they do that, they are stabilizing their ruminal fermentation conditions. Okay, so we've been spending a lot of time thinking about the prepartum and postpartum outcome, focusing on that low dry matter intake. In reality, what we've been promoting has changed over the last 15 years. If you look at the old kind of manual, the NRC for dairy, at least the one that came out in 2001, we would have been suggesting to feed higher energy diets prior to calving to pre-adapt that rumen environment to that greater starch concentration. We know that that ruminal adaptation concept is true. However, there are other issues that override potential benefit from that system. And so we know as we feed higher energy diets in the close-up phase, we increase body condition score prepartum, we increase the magnitude of the dry matter intake reduction, and we tend to increase body condition score loss postpartum. And so those are obviously negative effects. But if we think about this from a rumen adaptation perspective alone, we can see that once we feed a high energy diet, shown in the more dense dots in this image, we can see increases in ruminal papillae surface area, allowing potentially for greater absorption of those short chain fatty acids. And that's indeed what I show you on the right-hand side, where cattle that were adapted to a higher fermentability diet had greater rates of short chain fatty acid disappearance than those non-adapted cattle. So I was fortunate enough to do my PhD with Dr. Oba, and Dr. Oba has done some really exciting work in this area, focusing on the connection between the prepartum starch and the postpartum starch. And to summarize Dr. Oba's theory, his suggestion is that the ideal starch concentration in the fresh cow diet is directly related to the starch concentration in the prepartum diet. So in his opinion, and the concepts that he's promoting are that the starch concentration, if we have it high in the prepartum diet, it increases opportunity for cows to cope and tolerate with a higher starch concentration in the postpartum diet. So maybe not having a fresh group, moving directly into that high lactation group. Or if we choose to use the controlled energy program and we by default have a low starch, we may actually require lower starch concentrations in the postpartum phase to allow for that rumen adaptation that still needs to come to promote that short chain fatty acid absorption. There is some evidence for his theory, and Dr. Oba is a pretty conservative person when it comes to making bold recommendations. And so this is one of the studies that he conducted with his grad student at the DRTC. So in this study, they fed either high or low starch diets for 28 days prepartum. They then imposed rumen acidosis challenge models, 10 to 8 prior to calving, 7 days after calving, and 21 days after calving. And you can see that the higher the starch diet during critical phases, so within basically the last week prior to calving and the first week after calving, the cattle fed that higher starch diet were at greater risk for ruminal acidosis than those cows fed the lower starch diet. So it's highlighting again that increased risk for ruminal acidosis. They followed this up with another study, and in this study, they actually had 2 more treatments that I'm not going to share, but in this case, they had a controlled energy prepartum close-up diet, and they transitioned those cows to either a high starch diet of about 28% starch or a low starch diet that was about 22% starch. Okay, and they monitored this for the first 23 days of lactation. And so that's what I'm showing you here in the table. And is feeding that high starch diet did not drive greater dry matter intake. It actually reduced milk yield. So they produced about 2 kilos less milk per cow per day on that high starch diet when fed a low starch prepartum diet. And fat yield was not improved feeding that high starch diet. So although we might think we need to bridge that energy gap by increasing the energy density of the diet, we may not actually observe that big of a response, probably because of the changes in the rumen dynamics that are occurring, the potential for low pH to decrease fiber digestibility, and that energy cost that occurs if cows experience severe enough rumen acidosis to induce systemic inflammation. So there's other studies that have really looked at this response, either looking at a rapid or a gradual adaptation to a higher starch concentration. This is some work out of the Netherlands where they looked at trying to deliver either very rapid increases in starch to increase the energy density and energy intake by early postpartum cows. And you can see very clearly that dry matter intake was actually reduced in this case with the higher concentrate feeding approach or the more rapid concentrate feeding approach. And it also did not stimulate greater fat corrected milk. Here's the overall response in terms of dry matter intake. You can see that the gradual adaptation to the same concentrate level, but a gradual increase actually stimulated dry matter intake from a tendency standpoint, so a numerical increase, but there were no differences in terms of milk production. So obviously this is suggesting that with at least a numerical increase in dry matter intake, there's probably greater energy intake for those cows, and increasing that concentrate rapidly probably is not achieving the goal that we hope it might. Okay, so bringing this back to concepts that we should be thinking of, if we relate this again back to automated milking systems, we need to think about key indicators of a successful nutrition program for cows in AMS. So here are the three options I gave you: a well-established feed tables based on days in milk and level of production. Is that the factor that's going to drive a successful nutrition program? A low energy PMR to make sure that those cows actually have motivation to go to the AMS, or balanced PMR and AMS allocations? Okay, because I have control, I like the outcome here, so I'm going to end the poll and share the results. And what I've been really communicating over the last 4 or 5 years since we started AMS nutrition research is we need to quit focusing on the AMS feeding program and recognize that cows in the AMS require both the PMR and the AMS to meet their nutrient requirements. So I'm really excited to see that that concept is being promoted from those of you attending as well. Okay, so, you know, this really goes back to my theory behind dietary principles in AMS systems. Really, the cows have not changed. All we've changed is the feeding system. And so we shouldn't be thinking that there's this magical new approach. All we're doing is separating dietary components into a partial mixed ration, and then we're delivering some as the AMS concentrate. Our goals are the same, right? We're still trying to meet the nutrient requirements of the cow. I think the difference is, in this case, in automated milking systems, we're also trying to encourage voluntary attendance to that AMS system. Okay, so we're all in agreement of the importance of the PMR and the AMS. If we just now focus on the AMS feeding program, should feeding tables be designed to maximize pellet delivery for those early fresh cows, which is a common approach that I see? Should we provide a gradual increase in pellet availability, or should we rapidly increase pellet availability? Great. I think the responses are slowing down here, so I'll share the results. What you've clearly told me is we should be providing a gradual increase in pellet availability. So you may have seen some of this information already, but I think there is an overwhelming amount of data that is starting to come out. So there's still a very limited number of studies, but the studies that are coming out are having very consistent outcomes. And so I want to share those with you as we go through the last few minutes of the presentation. So we had the opportunity to collaborate with Dr. Oba and Jen Hayson from the University of Alberta. And Jen spent some time at our research facility in Saskatoon at the Rainer Dairy Research and Teaching Facility conducting this study. So she enrolled almost 60 cows. They were balanced between primiparous and multiparous, started them basically at a long close-up phase, so 28 days prior to calving. They all went through the same controlled energy diet prepartum, so 12% starch, targeting about 1.4 megacals of NEL, and there was about 1,200 grams of MP predicted to be delivered in these diets. They were housed in freestall, trained to use Instantech bunks so that we could measure prepartum intake. And then after calving, we moved them to the AMS and we assigned them to one of 3 different treatments. Okay, so they were either assigned to a low and constant amount of pellet in the AMS, so they only received 3 kilos for the first 28 days in milk, or we assigned them to a high allocation of 8 kilos per day, and they first received 3 kilos for 2 days, and then they either had a gradual increase to that 8 kilos over 15 days, or a rapid increase to the 8 kilos over 5 days. So we were looking at both the amount of pellet provided, but also the rate of that adaptation. Okay, this is our pellet. It was just a very generic pellet that had good pellet durability characteristics and reasonable palatability. 46% barley grain, 24% beet pulp, 19% corn grain, and then almost 10% molasses. Okay, if you look at the diet, what the nutrient contents were providing, the PMR was around 16.9% protein, 22% starch in the PMR, almost 33% NDF. And the PMR was programmed to allow for about 35, 36 kilos of ME and MP allowable milk. Okay, if we focus on how those cows could be accepting that diet formulation, assuming that they actually eat what's formulated, we would be targeting about 43 kilos of ME allowable milk and 44 kilos of MP allowable milk versus 44 and 44 kilos. So in theory, there's a small increase in the energy allowable milk for those cows that would be fed the high energy or the greater inclusion of that AMS pellet. Now, we know that cows don't always just eat more pellet. When they eat more pellet, they decrease PMR. And so we can look at a substitution ratio. And if they do substitute, obviously this means that the energy and protein allowable milk yields will be reduced. If they do not substitute much or have a positive substitution response, you can see we have a very positive potential production response coming our way. So this really was designed to test how are these cows in early lactation going to respond to increasing pellet allocation and whether the rate of that change impacts the response. Now we had some very surprising and unsurprising findings. As I'm sure you've heard me in the past, we can program very high rates of pellet allocation, but cows rarely can achieve intake of those high rates. And so even though we programmed those cows on the high allocation at 8 kilos, they were nowhere near consuming those 8 kilos, and they only consumed a little better than 4.5 kilos in the first 4 weeks of lactation. You compare that to our low allocation, and our low allocation outside of the first week, we're actually very close at achieving their targeted pellet intakes. So we already have a challenge coming at us in that the pellet allocations that were intended were not consumed by those cows during early lactation. We followed those cows for the next 4 weeks as well. So going all the way out to week 8, you can see they still did not consume those 8 kilos. And this is very common when I look at commercial data. You can see quite often that cows are not actually achieving their pellet allocation in terms of pellet delivery. And when we look at pellet consumption, there's even a greater discrepancy. In terms of PMR intake, no major effects coming from high or low pellet allocation. So remember, all these cows were fed the same PMR. And so we actually had higher PMR or numerically higher PMR intake for cows on that low allocation than cows on the high allocation. We did see a tendency for greater intake on the moderate adaptation protocol than on the rapid adaptation protocol. In terms of milking frequency, this is where the responses actually started to get quite surprising. Not significant, but a tendency for greater frequency of milkings for those cows fed less concentrate in the AMS, okay? And at least approaching a tendency for milk yield where cows fed less concentrate. Again, this is a guided flow barn in our AMS, numerically had greater milk yield. You can see the response is probably driven by variability. So if we look at variability in intake, our low pellet allocation consumed their daily quantity quite consistently throughout the study. When you compare that to either the gradual or the rapid adaptation protocols or feeding management programs, you can see the error bars are quite dramatically greater. And again, we're still not achieving that ramp that we had intended to with our feeding protocols. So again, it's more than just programming the concentrate in the robot, it's ensuring that the cows are actually achieving and consuming the program that is intended. Again, another surprising response is that our fat yield was actually statistically greater for those cows that were fed less pellet in the robot. Remember, at least numerically, they ate a little bit more PMR. And so we probably are delivering more energy through the diet. The reason I can argue that is because when we looked at body weight, body condition score change, plasma glucose, milk BHBA, and serum BHBA, we could not detect differences between treatments. So this really suggests that that low AMS allocation was not causing greater body tissue mobilization than the high allocation, and probably points to the challenges that I've been addressing, where if we try to target too high of concentrate allocation, particularly for those early lactation cows, we might be increasing risk for rumen acidosis. So just very quickly, I often hear, well, Greg, your responses are great, but you work in a guided flow barn, and many of us do not have guided flow barns. So the question is this a guided flow barn reality? Well, this is a study out of Denmark. In this study, they have a free flow barn. They tried basically a low allocation versus a higher allocation. Points I want to make again are the same. First of all, their high allocation never achieved their high allocation, and you can see the large deviation in the amount of concentrate that those cows consumed when fed the high allocation relative to the low allocation. We can also see that from a energy corrected milk perspective, there was no positive response of feeding that high allocation early in lactation or later in lactation. And in fact, they actually found lower milk fat and protein concentrations for those high allocation cows than the low allocation cows during late lactation. So I think all of this data is really pointing to the same message, that we need to seriously consider how we're designing these step-up tables and make sure that we're considering the PMR in that response. So major take-home messages: I think rumen acidosis is likely a challenge for our fresh cows. I think if we do have programs that increase risk for rumen acidosis, it may interfere with our goal of increasing dry matter intake and nutrient availability. And that's really the challenge that you identified at the start of this presentation as minimizing that negative energy balance. I think that tolerable ideal dietary starch is likely affected by the prepartum diet, as Dr. Oba has suggested. And then, as I've highlighted, feeding management with automated milking systems really probably is not that different than when using a total mixed ration. But with the PMR, we must consider both the AMS concentrate and the PMR in terms of supplying nutrients. And AMS feeding strategies aiming to maximize pellet intake during the fresh period probably will not be realized and may not achieve the most beneficial response. Thanks for the opportunity to have a discussion with you. I hope I have a couple minutes for questions, Jody.
Yes, you'll have a couple minutes, Greg.
Perfect.
So I'll ask everybody, if you have any questions for Greg, to please post them in the Q&A panel. And while we're waiting for those to come in, we do have a couple of questions that were sent to us early. So is it true that there's work going on with respect to an on-farm calcium test similar to a ketone test? And if so, when might that be available?
So that's a very good question. I'm not aware of on-farm testing for calcium. So I'm probably not the best person to answer that. Maybe Mark or Mike, have you heard of such a development?
Yeah, actually, there's two different groups that I've seen that are developing cow side testing for calcium, a rapid test.
So do you know when they're intended to come available, Mark?
I know there's a group out of faculty out of U of C that's doing some field testing, and I believe with some Lakeland College, and then looking at some kind of more field-based application, I think within the next 6 to 12 months. And then I had one of our tech people share with me a month or so ago, they saw a group on the US that was developing a calcium cow side test. So they were digging into when they thought that might be available commercially.
Thanks, Mark. And then another question for Greg, for close-up cows, what should we be feeding them so that they will not get milk fever?
Yeah, so there's a number of different approaches from a milk fever perspective. Obviously the DCAT approach has been quite common. We've done quite a bit of work. Actually, while I was still at the U of A, we were looking at strategies to enhance the negative ions, so the anion concentration in forages, to reduce the need for a formal DCAD program. I think probably the most important part of that work is it really showed that even a modest negative DCAD, so in that 0 to -5 range, was pretty effective of reducing risk for even subclinical hypocalcemia. So I think there's a lot of work still being conducted to discuss or debate how much you should acidify if you're using a DCAD program. The line of thought that I've been following is a mild acidification probably is effective enough and still beneficial for those cows. Now, if DCAD programs aren't what you like to implement, maybe it's feeding management or group sizes, maybe it's simply you don't want to be observing or assessing urine pH, then there are new products looking at calcium binders or utilizing calcium binders. There is some recent data that has come out of Cornell University showing a beneficial response in helping cows maintain postpartum ionized calcium concentrations. But I think at this point, really how you implement either strategy is a potential opportunity to control that milk fever incidence.
Okay, thank you very much, Greg. Unfortunately, we're out of time for questions, but please post any questions you have in the Q&A and Greg can get to them during the next presentation. Also, if you're able to stay around, we'll bring all of our speakers back at the end for additional questions.
Awesome. Thank you so much, Greg, for a wonderful presentation and some good discussion at the end. As Kira said, we're very fortunate to have the extra time at the end. If you're like me, you need to ruminate on that. And if you need even longer than that, don't forget we have our Ask an Expert, and this session is being recorded and will also be available on the DRECA web pages. So I'd like to introduce our next speaker, Dr. Mark Cameron, Vice President of Sales and Nutrition at More Than Just Feed. He enjoys being very hands-on there, whether it's on farm providing technical expertise or mentoring employees. He's always focused on what's best for the customers and the team. Mark earned his master's in dairy science from the University of Illinois and furthered his passion for learning by earning his PhD in animal nutrition of ruminants. Mark has over 25 years of experience in the industry and is continually looking for ways to improve contributions to customers. Thank you, Mark, for joining us.
Good afternoon, or I should say good morning, everybody. I first want to thank Alberta Milk for the opportunity to share with you guys a little bit today. I've been reviewing some research over the last 6 or 8 weeks. I also want to recognize Mike and Greg. We are truly blessed to have 2 world-class researchers housed in Canada that are committed to our business. So it's really cool to listen to them all of the time. Can you see my screen, Jody?
We can see it, just need to switch view setting, which I think you're about to do.
There, I think it. All right, so what I'm going to talk a little bit about today is fat, and really talk more about feeding fatty acids. And I'm kind of in a unique position today where I get the pleasure of listening to 2 really solid researchers. I'm working with producers on a routine basis and other nutritionists, and kind of in that hybrid model of trying to figure out what to do with this information. But if we think about feeding fat and we think about setting the stage here, is why might you choose to feed fat? Are you trying to increase dietary energy density of the diet? How does that support milk production? Or in your guys' case, really, how do we drive kilos of milk fat and the effectiveness and efficiency around that? And then there's also the back end piece of what does that do around body condition or recovery, and how does that impact immunity, health, and reproduction? Also, as I look at the research here, I understand that you're managing cows in group scenarios, realizing that we create average numbers for the herd or for a group of cows, but we still have all of this individual cow variation, and how do we make that work? I want to recognize that there's been a huge amount of published research in the last 10 years around fat and fat metabolism. And in fact, American Dairy Science Association, a couple of years ago, they had a whole conference dedicated to just looking at lipids and dairy nutrition from feed to milk fat. And guess what? There's lots of moving pieces. So the stuff I share today, we're dealing with biology. We're dealing with your individual farms and management and forages and grains that are available, and you have to keep that in context. I did have Alberta Milk pull this graph for me, is that if we step back to 2010, we can see what your average milk fat percent was as an industry here in Alberta. And then if we look at 2015, we can see here that it's increased. And then if we look at 2020 and look at the whole year, it averaged over 4.2%. And what's the driver of that? If we look at your genetic base and all of the changes you've made, we're driving kilos of milk per cow up every year. But then why are we also getting this corresponding increase in milk fat percent? It's really no mystery. You guys have embraced using fat at a feed ingredient level to drive animal productivity. So when that sits in my chair now, then what do I have to think about when I'm working with you at a farm level is, what are your different sources of fat or oil? How is that going to impact rumen function? How does that impact rumen biohydrogenation if that's occurring? And what are the actual fatty acids flowing to that small intestine? Some of the review I did over Christmas and in the last few weeks is really understanding there's lots of nutrient partitioning going on, and things are happening at a cellular level that really impact how that fat source you select may be utilized by the cow. And then if we go a little bit at what you guys deal with every day is, how do you deal with nutrient efficiency when you have fresh cows and high lactation cows, and you get cows at the later end of lactation, and you're dealing with milk production, reproduction, immunity, and then getting these feedbacks from the consumer about how do they feel about your dairy products. So lots of things to think about. I went old school and reviewed a paper on the 100 years of kind of fat research. And this guy Maynard in 1937 talked about that if we restrict the level of fat in the cow's diet, we see a drop in milk yield. And I went down here and I pulled a research paper, and I just looked at a group of cows. They were producing approximately 47 kgs of milk, 3.65% milk fat, which resulted in about 1.7 kilograms of milk fat that we were harvesting from that cow every day. In this trial, they averaged 21.1 kilograms of dry matter intake. So if we take our kilos of milk fat divided by our dry matter intake, is at a dietary level, we at least had to provide 6.5% of total fat, realizing that fat's going to come from different pools. It's going to come from de novo synthesis and how we digest forages at a rumen level. We have adipose tissue, or maybe cows mobilizing body condition and early lactation. And then one of those dietary fats we're feeding. But if we're going to make kilos of milk fat, we have to feed fairly high levels of substrates that drive that synthesis of fat, milk fat. I wanted to step back and just quickly think about, where was our industry 10 or 15 years ago when you were making decisions about how to optimize quota utilization? There was maybe a little bit more of a balance between the value of milk fat, milk protein, and a little bit on solids, non-fat. And then that maybe changed how you look at adding feed ingredients into your diet. We know that maybe we're going to cap out at about 6.5 to 7% total dietary fat. 2.5 to 3% is going to come from our base ingredients, and anything in brackets, that's kind of an average number. So alfalfa, hay, or silage would maybe have about 2% fat. If we look at rumen available fat, we were feeding a lot higher levels of corn distillers, and the refining process wasn't as good, so they were higher in fat. I saw a lot more producers feeding more tallow in their diets, bakery byproduct, occasionally some whole cottonseed, definitely some roasted beans in Manitoba. And then kind of the third pool of fat was always these inert fats, something like a Megalac or Energy Booster, but because of price, maybe only feeding 200 to 300 grams per cow per day. Now, if we step into today's environment, we see a whole wealth of knowledge around feeding these inert fats. We have more options for calcium salts. We have more options around prilled fats. And then we see these enriched POM fats that are greater than 85%, tons of product offerings out there. And then throw in a little bit of these inert fats that have been developed to enhance reproduction. You guys have a lot more choices about how you want to drive effective kilos of milk fat. And just a quick slide here. I mean, these are the kind of products that are available in Western Canada. We have our roasted beans up here. We got some whole cottonseed. But if we look at all of these commercial fat sources, you got a lot of things to think about on what you pick and how they might impact lactating cow performance. The reality is you guys have an economic engine and you're looking at how do we drive kilos of milk fat. And as I go through the research here today, I want you to think about it, that kilo of milk fat is a combination of two interactions. How much milk volume we're going to produce per cow per day, and what is that milk fat percent? But it just doesn't stop there. How does that impact your quota utilization? Do you lease quota? Incentive days, all of those things that might impact how you choose to feed your cows to drive your revenue at the farm level each and every day. So if we just do a quick look at milk fat percent, what drives it? We're going to see really three streams of fatty acids that drive that milk fat percent. We have de novo fatty acids, which are a shorter chain C4 to C14. That's made at a mammary gland level. It's really impacted by how we digest forage and grain at that rumen level. It's all about rumen function. However, there's a fair bit of range when you look at published data on that de novo fatty acid profile, anywhere from 18 to 30%. If we look at our preformed fat, this is fat in the diet, or maybe being mobilized, that body condition adipose tissue, and it's 32 to 42%. And then we have this mixed fat, which is really heavily based on C16, and it can come from both streams, some made in the mammary gland, as well as some coming from the diet, but ranging from 30 to 40%. So the reality is when you synthesize milk fat, there's definitely different streams it can come from with a fair bit of range and how that animal might respond to the different feeds you're feeding. If we look at
Mark,
dietary...
we have a quick question.
Sure.
So the question coming in is, will feeding high levels of fat affect reproduction?
I looked only a little bit at it, but yes, if we pick some essential fatty acids, we can influence reproductive success around first service conception rate and how many cows will get pregnant by 100 days in milk. The time I spent looking at that, though, is we need to look at different ways of feeding that fat. It's kind of what we do the first 3 to 4 weeks pre-calving. There might be a specific essential fatty acid strategy for the first 30 days. And then you might actually change to another type of essential fatty acid post 30 days in lactation to really drive reproductive success. So great question. And there's some really solid data evolving out there that we can drive reproductive success. However, it's really a function of how you can group cows successfully on farm to take advantage of that and be cost effective. That's the key. Being cost effective with a strategy. If we look at feeding dietary fat, really I'm looking at 5 main fatty acids. We have 2 saturated, our POM and our stearic, so a C16, C18. Then we have our unsaturated, 18-1 oleic, 18-2, and 18-3. And if we take a look at things from a dietary basis, this is at a high level. If we were to feed straight tallow, we see here that it's pretty high in POM fat, stearic, and oleic. If we look at traditional calcium salts and they're in that inert form, they're really a blend of really POM and an 18-1 oleic. If we go to something more like an energy booster or a dairy natural vitality, that Prilled saturated free fatty acid, they're a blend of POM and stearic. And then if we go to these enriched sources of POM fat, yeah, they're really super concentrated. Sometimes being in that 85% range, but frequently being 90 to 95% POM fat. So we got to maybe stop thinking about fat as fat and thinking really about what's the fatty acid profile of whatever we're choosing to feed. I love this slide that I've seen Adam Locke from Michigan State present a number of times, is as we get to the producer level, we have to think about what you feed this cow, what happens when it hits the rumen, and ultimately maybe what's being pushed into that small intestine, and how we affect fatty acid digestibility, and how much fat, how many individual fatty acids are being absorbed. Realizing that it's going to be impacted by if a cow's gaining or losing body weight. And that's really going to impact then the synthesis of milk at the mammary gland, but it's just not only about making milk fat percent, we have to think about how we maximize maybe protein percent in yield, as well as lactose, because lactose is a driver of that milk volume. So I always applaud dairy producers. You guys really got to think about the complexity of nutrition. How do you simplify it within your day-to-day environments and drive cow efficiency? So we're going to spend a little bit of time looking at some research here now. What I'm going to do here is look at a trial back done in 2007. Its base forage is alfalfa hay and silage, some steamrolled corn. But what they did is they fed these cows either 500 grams of palm fat, 1,000 or 1,500. This product is actually our Energizer RP10, about 87% palm. And we can see the diet here increasing from 2.3 to 3.1 to 5.2 to 7.2 fatty acids as a percentage of the total diet. And then let's look at calorie response. On the 500 gram cows, they actually consume 476, 887 grams on the 1,000, and 1,248. If we look at dry matter intake, a little bit of a quadratic effect. The 500 grams showed an intake response. Again, off of one study, it says it's significant. Not sure if it's very real, but that's what happened here. On the 1,000 and 1,500, no change in dry matter intake. But we see a nice response on kilos of milk fat going from 31 to approximately 34 on any of the 3 fat treatments. If we look at milk fat percent, 3.44, and then it increases to about 4%, 4.1, and 3.9. And then looking at how you guys get paid on kilos of milk fat, a really nice increase in grams of fat being produced by putting that palm fat in the diet. Now, I tried to take this and do some cowboy math on it. And this is what this slide shows here, is on the cows that were receiving or consumed the 476 grams of the enriched palm fat, is they produced an extra 286 grams of milk fat. And all I did was take that, divide it by 100 grams or 4.76. And when this cow consumed the 476 grams, for every 100 grams of palm fat they consumed, we got about 60 grams more milk fat yield. Just trying to put something into a simple, practical measurement. And if we go to the 1,000, then it's 34, and the 1,500 was at 32 grams of extra milk fat for every 100 grams of milk fat consumed or palm fat consumed. And again, just trying to look at something about efficiency here. What is the sweet spot when we start looking at putting inert fats in the diet?
Mark,
what happens?
we have another question.
Sure, go ahead.
The question is there a difference in utilization between prilled and flaked fats?
So this is a flake fat. This study here didn't report any digestibilities, so I'd be making a number up. On the data I've seen, I don't think there's a whole lot of difference. However, here's a statement that I will share as we walk through a couple of these trials. As we increase the amount of inert fat we're feeding per day, we are going to see a drop in that digestibility. However, it's the amount of grams of fatty acids we get delivered in that small intestine that can help drive productivity. So hopefully that answered their question. If we look at these diets though, and we look at the fatty acid profile in the milk, we can see here that C16 increases from the control diet as we go across and put more palm fat into the diet. And the de novo percent starts to go down. And guess what? The ones that are in red here, it doesn't concern me as much as I thought it would, because we're driving so much more intake of fatty acids that we end up with a very effective production of kilos of milk fat. Here's a study that looked at the longer term, and I won't spend too much time on the diet analysis here. I just want to show you that the base diet was about 3.4% fatty acids. It goes up to 4.85, so we're adding about 1%. Again, the amount of C16 was 81%, because again, it's a palm fat diet. And if we look at cow's production here, from calving to 10 weeks into milk, we can see a nice response on dry matter intake on these cows consuming about 458 grams of palm fat a day. And if we look at milk fat yield, we see a similar response. These cows were very effective at creating an increase in milk yield. And if we look at the data here now, we have really good intakes on these cows. The cows that were fed the palm fat for that 10-week period, about 4.3% of body weight. So really good at driving intake. And because of that, we had higher kilos of milk being produced per day, 49 versus 46. Our milk fat percent went up from 3.2 to approximately 3.4. And that drove about another 150 grams more net milk fat per day. And again, if I look at the amount of fat being consumed, this palm fat, for every 100 grams being consumed, I got about another 33 grams more milk fat. And what was interesting here, I drove volume of milk, I drove more kilos of milk fat, and guess what? Those cows on the palm fat showed a trend towards being more positive in change in body condition score, actually gaining some weight. When we take a look at this from a diet intake and total tract digestibility here, what did I see? We got more total fat intake on a grams per head per day basis. What is interesting on this work with the inert fats and the palm fats especially is we see an improvement more often than not in total dry matter digestibility and NDF digestibility. So here, dry matter digestibility being about 66% on the cows fed the palm fat versus 62, and increasing on NDF from 45 versus 40, and getting more absorbed fatty acids in those cows through these enriched protected palm sources. Here's another study looking at maybe using two different fatty acids from an inert perspective and having them protected and being blended, using something high in C16, but also adding some C18, that oleic into it. And they fed very specific ratios of the palm fat being 80% in one diet and 10% of the 18-1, all the way to being down at 60% of the C16 versus 30 of the 18-1. Now, what does that mean at a fatty acid supplement on grams per cow per day? Is on the diet that was 80-10, the cows are getting about 400 grams of an enriched C16 and about 50 grams of a calcium salt. All the way that we had a big shift that we got 141 grams of a C16C enriched and 360 grams of a calcium salt. And if we take a look at it here, if we look at energy corrected milk, there's really no differences across these four dietary treatments. We do see some changes in milk fat percent, 371 when we're feeding higher levels of the C16, and drops down to about 3.5 when we feed a diet that has more of the C18-1. However, no difference in kgs of milk fat. So I think we can use these inert milk fats, or use these inert fatty acid supplements with different fatty acid blends if we understand what we want to do, and how do we want to drive that kilos of milk fat. Is it around milk fat percent and volume, or we want to drive more volume and maybe with a little bit less of a little bit lower milk fat percent? And then if we look at body condition changes down here, as we put this C18-1 in, some improvements in body condition and how these cows are gaining over this study. What was interesting in this study, and what I really like about this, is they took this pool of cows that were fed these four different fatty acid strategies and really broke them into different production groups. The low group were averaging 45 kgs of milk. The medium group was at 53, and the high group. And you can see here on milk yield, there's a linear significance to what happens on these cows when we feed different fatty acid profiles. And when we fed something higher in the C18-1, driving more milk yield than when we were on the 80%, 10% ratio here, where it's heavily C16 based. And if we look at milk fat yield, yeah, we saw some linear trends between the low group and the high group. So really thinking about what do I want to do with my cows? Can I group them? And can I use different fatty acid profile strategies to drive cost-effective kilos of milk fat yield? This is a study that looked at two components recently being published here out of Adam Locke's lab. And what they did is they looked at what happens when we feed palm fat to fresh cows. And then they also looked at then cows past 26 days to about peak milk yield at 67 days. So if we look at this here, we have a control diet. We have a diet with palm fat. And what they're doing is they have a diet that has a base level of fatty acids of around 3%. They're going to 4.5. So we're feeding palm fats at about 1.5%. And then the same thing when we went into this diet after 24 days in milk. Again, the base diet control was 3.6, and we're going to about 5.1%. So we're increasing and feeding 1.5% palm fat. What did we see here is if we look at fat yield in this 3-week period, the cows that were on the palm fat, we had a significant increase in milk fat yield, roughly 2.3 to maybe about 2.1. If we look at then between 4 to 9 weeks, we saw cows that received palm fat. This dotted line here, these are cows that received palm fat in the first 24 days. And as well as it continued on, we see this nice increase in milk fat yield. We took control cows and then added palm fat in the diet. And guess what? They responded and made more kilos of milk fat. So clearly a nice response to how those cows are utilizing that enriched palm fatty acid source. And if we look at the data here as a summary, really solid intakes at 22.1 kgs. We see this nice increase in milk and energy corrected milk. We see more milk fat percent. And then we see this nice response on kilos of milk fat. When cows are fed a moderate level of palm fat, averaging about 323 grams. And again, when I kind of do my cowboy math, I got 87 grams more milk fat for every 100 grams of palm fat, realizing that's a little bit misleading here because we got cows mobilizing body condition too. But between what we did from a dietary strategy, doing a good job of driving intake, and then using the body reserves they have, we can really drive high kilos of milk fat that first 3 weeks post-calving. And if we look at digestibility, we see improvements of dry matter digestibility and NDF digestibility in that 1 to 24 days, as well as the 26 to 9 weeks on those cows that are receiving the palm fat. So that's really exciting when I think about it from a nutritional perspective, that we're getting this lift in metabolic efficiency, driving more kilos of milk fat, but we're really getting good diet digestibility also. If we look at the data here on the cows from 25 to 67 days, we can see here that we got really solid intakes. As a percentage of body weight, all of the cows are in that 4.3 to maybe 4.5 range. On the cows that receive the palm fat, on these two columns here, we can see nice increases in kilos of milk being produced. That translates into more energy corrected milk kilos per cow per day. And then we see this response on milk fat percent being about 3.7 on the control cows or cows not getting the palm fat. It goes up to 3.9, 3.8. But again, driving these kilos of milk fat. And then when I kind of do my cowboy math and looked at how many grams for every 100 grams of palm fat they were consuming, how much extra milk fat we got 52 grams on the cows that went from the control to the palm fat. And then the cows that stayed on the palm fat the whole 9 weeks, they're at about 36 extra grams of milk fat for every 100 grams of palm fat. And again, to me, not super high levels of palm fat, 465, 447 grams of palm fat being consumed per cow per day. If we look at milk fat profile now, if we take a look at what happens on grams per 100 grams of fatty acid, this de novo number drops from 25 to 22 to 22. And a year ago, I would have thought that bothered me when I was doing some milk sampling of fatty acids at a farm level. But I realized as I reviewed this data, if we do a good job of holding intake, we drive rumen function and we're feeding these inert fats, some of this thought process about maybe what de novo percent should be kind of goes out the window, because if we come down here and look at grams per head per day on de novo, it's very similar across all 4 dietary strategies. However, when we put the palm fat in the diet, we're clearly getting an increase from roughly 590 to 720 grams per cow per day. And then that formed fat, the dietary fat, maybe the adipose tissue is very similar. So yeah, we can start to think about fatty acid supplementation differently. And we're really going to need to start thinking more about that fatty acid individual profile and how can we use that to influence how we make decisions to drive revenue as you guys harvest milk every day.
We have another question, Mark.
Sure.
It says we use C16 fats for manipulating production according to market demands, incentive days, etc.
Yep.
Is there a strategy you can recommend for bringing cows onto and then away from palm fats?
Oh, I think we had a brutal lesson in this spring when we pulled fat out of our diets and we lost kilos immensely. I saw producers that would take 200 to 400 grams of palm fat out and we would lose 2 to 4 tenths of milk fat immediately. And then we saw cows lose a lot of body condition and early lactation, and then maybe struggle a little bit with some reproductive success or failure in late spring and into those summer months. So it's really hard. As I've been thinking about this, my thought process is maybe what I would do at a producer level is if I'm feeding a lot of palm fat and we're driving this efficiency between milk fat percent and milk volume, is maybe we shouldn't pull all the palm fat out. Maybe we should shift to a little bit more higher C18 inert fat, give up a little bit of milk fat percent, but maybe keep our volume better on those fresh cows, keep them milking well so that when we have the opportunity to turn that cow back on with incentive days and I throw the palm fat in, we've already built the milk volume there, that kilos of milk per cow per day, but now I can supercharge her to drive more milk fat percent. So one of the things that I talked about with some of the consultants I work with that are working with producers every day is I think we learned we can't pull all this fat out. Because then we get cows in mid and late lactation, we can't turn back on the way we want. So we have to think about what we're doing and maybe design different diet strategies to kind of turn cows off and on more slowly, realizing that it's just hard to do. You guys know it, you live it every day. You just can't turn cows off and on as easy as we'd like to think about from just a straight science perspective. In that. Again, this study demonstrates what I definitely see a pattern in with these enriched palm fatty acids is an improvement in dry matter digestibility and NDF digestibility, which is good because that NDF digestibility is going to help drive the de novo synthesis and help keep those grams of de novo being produced at a higher level. So yeah, like it or not, the research kind of tells us that this inert enriched palm fat or some of these blends definitely allow us to influence how we're going to produce kilos of milk fat every day. And that's a huge driver of your revenue as you look at filling that quota every day. So as I finish up here, I know I covered a fair bit of ground here. It was kind of high level, but these are some of the things I think that you need to be thinking about as you walk into not having incentive days right now is why are you feeding fatty acids? If it's to enrich the energy density of the diet, is that because maybe we're not maximizing dry matter intake, or maybe our forage quality isn't quite as good as we want, but we still got to figure out a way to maximize that cow's nutrient intake every day. So as you engage with your nutritionist, start thinking about where do I want to be for body condition, either gain or loss as I pull fats in or out of the diet. What is my goal around milk yield, milk fat percent, and then think also about that, about how does it maybe impact some other component yields. One of the things I kind of gleaned when I was reading on these enriched C16 diets is maybe there's some glucose sparing going on that glucose is then being pushed to lactose synthesis, which will help drive more milk volume. So clearly you've got all of these interactions going on at a nutritional level. It's your fat, your fatty acids, it's your starch and type of starch you're feeding, the type of forage you have in the diet, and the digestibility of that, all driving all of those decisions that you guys have to make every day. I did look at some stuff from Dairy Science abstracts and kind of looking at a summary of some high-level analysis. And on 30 peer-reviewed publications on when palm fat, this enriched palm fat, was fed in the diet at less than 3%, is there's really no impact on dry matter intake. I showed you guys some data today that says it's no impact to maybe a little bit of an improvement. We're clearly going to drive milk fat yield, and we're going to drive milk fat percent. And more often than not, that milk fat percent increase is maybe 0.2 to 0.4 units of milk fat percent increase on the kind of diets we feed in Western Canada. Maybe a little bit of a loss of milk protein percent, but kilos of milk protein look really solid. What clearly came out of all of this peer-reviewed stuff on this abstract is that when we feed higher amounts of this enriched inert palm, we see an increase in dry matter digestibility and NDF digestibility. If we look at the calcium salts, again, feeding it at less than 3% of the diet, at times we can see a little bit of a decrease in dry matter intake. And I only can relate this maybe to a little bit when I was a grad student. We were doing some work with calcium salts at the University of Illinois. And often that decrease in dry matter intake was associated with cows that are on lower levels, and then we rapidly transition them to a higher level. And if we can manage that, gradually increase the grams of calcium salts, maybe we'll minimize that loss of dry matter intake. But even calcium salts, when we look at this reviewed data, strong signals around increasing milk fat yield and driving it through, and then that drives milk, or I shouldn't say, clearly we're increasing milk yield. We maybe don't get the same milk fat response, but we still get a good response on milk fat yield because of that combination of nutrient partitioning and how we're driving the production of milk volume with an okay milk fat percent. And again, a little bit of a reduction in milk fat percent. And here, not the same kind of degree of response, but still some consistent response on NDF digestibility for that total diet. So now, thinking about it from your guys' perspective, is you guys have lots of interactions you have to think about every day to make your cows be efficient. What's that blend of nutrition and management? When we start thinking about putting fat into the diet, how does that impact the type of forage or forage quality you have? What kind of ration or diet design, your feed delivery system? Greg talked a little bit about that. As I look at this data on the inert fats, what I saw more often than not on when it worked well is because those cows as a percentage of body weight had really, really good dry matter intakes. And you guys know that. The easiest way to drive milk production is to drive high dry matter intakes. Then if we can have good quality forages there, we can manage our starch load on those cows and we put the right fat in there, man, I think we can make cows metabolically efficient and drive lots of kgs of milk fat. The research does tell me, though, that to take the full advantage of your fatty acid strategy is, can we group cows effectively so we can target these individual fatty acid profiles to get the responses we want? So can we have a fresh cow group? How do we manage a high cow group? Can we have a mid to late lactation? Those are the things that you guys have to figure out at your level and how to keep your herds as efficient as possible. Also, I need to be cognizant of that we see a lot more on-farm production of I'm going to call it a blend of protein and oil-based feeds, where we have extruders, we're crushing or extruding soybeans and canola. So you have a product that is higher in protein and fat. And what does that do about maybe ruffle, and how does that impact maybe the result in a little bit of a loss of milk fat percent? What I do know is the data is pretty consistent. If we feed higher amounts of fat in the diet, including inert fat, fatty acid digestibility is going to go down. So we have to think about what's the right level of total fat to feed so we don't end up screening it out in the manure. I'm really excited about the research that's evolving here, looking at these blends of C16, 18, and 18-1, and the ability to alter nutrient partitioning so that we can have a blend of driving milk yield, milk fat percent, and then kilos of milk fat How does that affect body condition over time? How does it affect reproduction? That one study I showed you, that if we got a really good group of high producing cows, can we use a better blend of C16 and C18 to drive more kilos of milk and control body condition on those cows? So to kind of finish it up here, we're going to feed palm fat because it helps you guys meet your quota. That's the reality. But what's the sweet spot? It's going to be different for every herd. If we look at some published data, I think on fresh cows at 1.5 to 1.7% of body weight, that 300 to 375 grams of palm fat seems to be a really good spot, as long as we can drive intake and we got good quality forages. On a high group, that data, yeah, we saw really good responses at 1.8 to 2.3% of body weight, which means we're going to target those cows at about 450 to 600 grams of palm fat per cow per day. The fact is, feeding fat drives your kilos of milk fat. I'm absolutely convinced that after reviewing all of this research the last 6 to 8 weeks, that you can drive your kilos of milk fat by having a good, effective, inert fatty acid supplement strategy. And that's what you got to look at. You guys got to look at, how do I maximize my income over feed costs? So with that, I want to thank you for your time today. It's a pleasure to be associated with Alberta Milk and the producers that are part of that. So thank you.
Thank you very much, Mark, for your presentation. So we will get to some questions in a minute, and I'll invite our other two speakers to turn on their videos and unmute again. But first of all, I'd like to say thank you very much to all three of our speakers for your time and expertise today, and for making our first virtual DRECA workshop a big success. So thank you very much. All right, so for our participants who are able to stay, we will allow for some more questions. So first of all, I'll just go for the last few questions that were posted for Mark. Producers need to be concerned with the quality of milk they're producing. We know that high C16 diets are increasing the FFAs in the milk that's being shipped. BC and Quebec are measuring the FFAs in the milk that's shipped, and they're finding that FFA is high and processors are not wanting that milk. How do you suggest on holding production while producing a product that processors still want?
Hey, great question. And we've actually been reviewing a little bit of work on these free fatty acids in milk. A lot of it's European data because they've had AMS systems longer than we have. Nutritionally, I think there's a component there, but there's also a lot of other factors would influence the amount of that free fatty acid in the milk. Receiver jar, length of the lines that milk's moving, going through, the abruptness of how it's being handled. And when I actually reviewed this research a couple of weeks ago, because I saw some stuff that BC Milk had sent out, is that if we look at free fatty acids in milk at the time of harvest, that will be one number. Let's say it's 2, just for a number's sake. 12 hours later, 24 hours later, that milk's been harvested and it's starting to oxidize. So that free fatty acid number goes up anyway. And the data I reviewed with one of my colleagues a couple of weeks ago is the published data is not even sure what a low fatty acid, free fatty acid number might be. So I'm aware of it, but I think it's a lot more complex than just what you feed. Herds that are on AMSs tend to have a little bit higher free fatty acids because of how that milk's being harvested and the physical distance and how it's being handled before it gets into the bulk tank. I think we need to be aware of that. I did have a slide here that I didn't share, is that the end users of your dairy products have different needs. And I'm going to take butter as an example. If you're a baker and you want a nice flaky pie crust, you want higher levels of C16, that more saturated, because that butter is going to be harder. If you're a housewife and you're trying to spread that butter on your toast, you might like a little bit lower levels of C16 from a spreadability perspective. And the person making a cappuccino, wanting more foam on that milk, wants a different kind of product. So I think there's lots of opportunities to think about targeting your end product and getting it into a consumer stream that really appeals to what that consumer wants. So I don't think there's any right or wrong answer here. I think we need to be aware of these free fatty acids. but I think it's more complex than just saying stop feeding palm fat. There's a lot of things that influence that. But we need to be very cognizant of what the consumer wants and how they view the products that they purchase from our industry each and every day.
Thanks, Mark. So for anybody else, please feel free to post your questions in the Q&A panel and I will get to them. Also, if you'd like to ask your question live, you can press the raise hand button and then I'll be able to unmute you so you can ask your question. And finally, if you're joining us by phone, if you press *9, it will also raise your hand so that I can let you speak as well. So the next question we have is for Mark. So if we're feeding over 800 grams of palm fat, do you have concerns that it can stick to the organs and cause major liver damage? As well, will excess fat just pass through the intestine and leave the cow unused?
So just a couple thoughts on that. For those that know me and I've been on your farms, you know I love to screen manure. When we're pushing 800 grams of palm fat in a lot of diets, I think with the decrease in fatty acid digestibility or absorption, we do see some more of these prilled fatty acids show up in the manure. I also think at 800 grams, if we're getting that into a cow in later lactation, she is going to push that to body condition. And it's only because I've worked in the feedlot industry a little bit to know, and Greg is a great resource on this too, that if we start to lay condition down on that cow, it's not only what we see over the hooks and pins, we're going to start to lay down some internal fat, and we're going to see more internal fat. We might see more seam fat if they're truly fat. And then that concerns me on cows that are going to go into that close-up pen. That I looked at some data a few months ago and listened to some webinars on these cows that are maybe 3.5 body condition or more, that drop in intake probably occurs sooner pre-calving. So maybe instead of that stuff that Greg showed at 7 days having a big drop, maybe those cows are losing intake as much as 10 to 14 days pre-calving. Greg, any thoughts on that?
Yeah, you're right on with what I've seen. Obviously, that body condition mobilization also increases risk for other disorders. I'm right with you.
And then those fat cows with low intake pre-calving, I think there's some pretty strong evidence we get a lot more tag buildup in that liver pre-calving and probably post-calving, which doesn't help your whole subclinical clinical ketosis challenges in getting those cows onto feed.
Thanks, Mark. The next question that's in the Q&A, what are your thoughts on the future of palm fat? Would a local heat-treated whole canola or flax meal product work for our diets?
At a high level, as long as you guys are paid to produce kilos of milk fat, you figured out how to do it the most cost effective. The real push will maybe come from the consumer. So I'm not opposed to it, because when I review this fatty acid research and I look at the amount of palm fat, the cow makes milk fat and the fatty acid profiles, and they're based on the diet we feed. And everything I showed you today it's all falling within normal parameters around that de novo, the mixed and the preformed fat. Do I think it alters some dairy product characteristics? Yes, absolutely. Till maybe we can isolate milk into different silos at our processing plant, you're going to be blending product together, and it's just hard to do that. Now, I also think there's some opportunity there on some of these cold press or extruded canola meals, soybeans, and linseed out there. Yeah, and there's some work coming out on a recent product or a product in Canada called Limpro R that clearly looks at helping to drive reproduction and a few other things. So yeah, absolutely. I think things we should be exploring and looking at.
Thanks again, Mark. So again, if anyone has any questions, you can put them in the Q&A or raise your hand. Otherwise, I'll just go to some of the most upvoted questions that were answered by chat, just to make sure that everyone can hear the answer. So for Mike, When feeding the second meal at 12 hours, is it meaning the second milking of the cow, or is it more beneficial to source first milking quality colostrum?
Yes, based on the study that we did at the DRTC, I would be recommending even up to 12 hours feeding that first colostrum from the cow, even if it is at 12 hours and the second meal or even the third meal. I still think within that 12-hour range, especially, and arguably, and we don't have enough research to back this up, but I would expect even the first 24 hours, I would be feeding the highest quality colostrum you have.
Great, thanks, Mike. And then there's one more here. My calves really like this grass that I put out for them. It seems to go for it more easy than the starter, and this is pre-weaning. I limit the grass so that they eat more starter. Is too much grass a concern? This is high quality, high palpable grass.
Yeah, my first comment is that when you put out a section of grass hay, it looks really large, but if you weigh it, it's actually not that much. And I think that although it looks like they're consuming a lot, when it comes to kilogram dry matter intake, it's probably a lot smaller than what we think. So it's good to feed it. I have no problem feeding that. There's some work out of University of British Columbia showcasing that if you do feed a higher quality hay to your calves, you're going to have less starter intake. But when you look at the total energy intake and the total growth, it's the same. So the calf has a way of regulating its energy intake based on some of the slides that I was showing you today. It can happen in the milk feeding, but also in the dry feeding as well. So I would say there's no harm in that. That's not a problem. I don't think it will be depressing starter intake to a level where it would be depressing growth. Because when it's all said and done, they're going to be consuming far less than 20% forage at that period.
Great, thank you very much, Mike. For Greg, in the AMS, does pellet size make a difference in how fast cows can consume the pellet?
Yeah, so that's a great question. There's not a lot of work done in AMS systems on pellet size. We know if you feed a pellet versus a mash, there's very big responses. And this data is pretty old. It's going back to the mid 80s or early 80s when it was investigated. But it's suggesting that cows can consume about 450 grams of pellet per minute. And that drops down to around that 300, 350 grams per minute if you go to a mash. Now, I know there's a lot of producers that are interested in using homegrown feeds. And so if that's of interest, it doesn't mean you can't use those homegrown feeds. You may just have to rethink your feeding tables within the AMS program. Getting at the pellet size question directly, I think we need to recognize that AMS systems do not measure the amount of product that they're delivering. And with larger pellets, there could be more challenges maintaining adequate durability. So those pellets may break up more during augering or during handling. And if you have more fine production, that may increase refusals in the bottom of the feeder at the AMS, or it may create a more variable amount that's delivered because that AMS is just dispensing based on the number of auger turns. So pellet size, I think, is something to play with, but pellet durability probably trumps pellet size for AMS systems.
Great, thank you. We have a couple of new questions. So for Mike, does a second milking that has a high Brix number qualify as good colostrum? How does one get cows to give more colostrum?
Yeah, so the first question, if the Brix is over 22, I would say, and it's even the second milking, I would classify that as high quality colostrum. So you can definitely feed that. I would just use that cutoff of 22, seems to be the most effective. I know that a lot of other farmers are trying to push the boundaries and always get around 28. But I think that the 22 would be acceptable in that circumstance. And the second question from Garrett was, again, how do you get cows to produce more colostrum? That's an awesome question. And unfortunately, we don't really know the answer. We know that seasonality has something to do with it. We also know, and it looks like that energy prepartum has something to do with it. So that's kind of ironic that you're asking that question, because that's a big have a lot more answers, hopefully, fingers crossed.
Thanks, Mike. And yes, I'm sure we'll be inviting you back.
Yes.
For Mark, if you're still there, would a high-fat canola meal replace palm fats?
It can, and based on some field samples I've seen, if we see these high-fat canola meals that are coming from a cold press, and they're in that 12 to 15% range, my biggest fear there is how fast that oil is released at a rumen level. That if we drive some CLA production, that doesn't take much to really drop milk fat percent synthesis. And just to put it in context on some data that I had a chance to look at when I lived in Ontario, is that 20 grams of a protected CLA, if you fed that into a cow and they're at a 4% milk fat, it would drop it to 3 in less than 24 hours. So that's the bigger concern I have on a canola meal or a flaxseed meal or a soybean, is how much oil's there and how quick that gets released at the rumen level, and maybe drive some CLA production that you all know from 15 years ago really can play havoc on milk fat percent. So can you use it? Yes, and we have producers using it. You just monitor a little bit more and think about the level you're feeding to make sure we don't cause any milk fat depression.
Thank you. Another question for Greg. What's the best pellet delivery feed in robot feeding?
I'm not exactly clear what the question is getting at in this case. Is it possible for Leighton to clarify?
Let's give Leighton, if you could rephrase your question. Oh,
yeah. So Leighton now mentioned speed. This is I think a big debate. Usually we try to target feed delivery rates at the same rate that cows are consuming them. So the main reason is if you deliver at a rate that is faster than that, so above 500 grams per minute, there's a high likelihood that she may not consume that whole meal. And when she leaves the robot, there'll be pellet left behind for those cows or for the next cow. If you feed at a rate that is far slower, then the argument becomes that she may get frustrated with not having sufficient feed delivery. Now, there's not a lot of data that really supports that. And depending on the type of system you have, it may automatically adjust the feed delivery rate. So for example, the Lely systems adjust feed delivery rates based on milking times. Whereas if you look at the D-Level system, we can set a specific feed delivery rate for individual cows using the herd as a whole average. So there's no real good data saying what's the best. I think the logical approach is to target a rate that allows for sufficient intake given the milking times, considering they're probably not going to eat much more than 500 grams a minute.
all right. Thank you, Greg. I will just mention that we'll plan to wrap everything up by 12:30. So we'll just get to our last few questions. So for Mike, would feeding more than one meal of the first colostrum have a different impact on GI tract function or improved function compared to feeding one meal of colostrum and then transitioning to a whole milk or milk replacer?
Sorry about that. I unmuted myself. I'm not sure if it will have an effect on gastrointestinal development or function if you feed that second meal of colostrum up to 12 hours. I would expect that if you feed a little bit more energy in the first day of life, you'll probably promote a little bit more development. But where it becomes very clear to me and why I'm recommending this is the bump in immunoglobulin levels in the blood from not just this experiment, but other experiments when we're feeding high levels of colostrum in the first hours of life. And if the gut is still open at 12 hours, which it appears to still be, I would just keep on pumping the calf full of immunoglobulins and other bioactives that it needs. Now, this could be short-sighted, and we definitely need to do a lot more research in this area, looking at those transitions, and that's a big phase of our next round of colostrum research, is to look at that transition period. But for now, just in the bump in immunoglobulin levels, I would still go for that. And unfortunately, I just don't know if we're going to be affecting the gut. But that was an awesome question.
Thanks, Mike. Mark, we have another question from you that came in externally. With the different types of POM fats out there, are some that are better than others? And what characteristics do you look for in a POM fat that's better?
Well, when suppliers come to my office, one of the things they need to do is they need to bring research specific to their product. Then if they provide me some product, we're going to analyze it to see if what they're recommending is the fat level and the fatty acid profile, is it showing up in their product? So frequently, I'll get 4 to 6 samples from over time from a supplier and then actually analyze them. So I want to know it's research based. I want to know consistency of the product. I need to know that it's inert so that we can get it to the small intestine so we can drive the productivity we want out of it. Yeah, I think there's a lot of good products on the market today. So I'm not going to talk about any commercial brands because I think there's just a lot of good products out there. And you can see on my presentation, every one of the trials, I let you see what the product was. So yeah, I think there's a lot of good options out there. It is about research-based, and how consistent that product is from a fatty acid profile is how I make decisions.
Great, thank you. So for Mike, what is the minimum liters you would feed from day 2 to day 7?
So a calf right now, entering this weather in Alberta, just to let it survive, no growth, 6 liters of really concentrated milk, probably 7 liters to be more conservative. That would be the minimum, I guess, during really cold temperatures. Maybe on more thermal neutral times, it would be a little bit lower, around 5 liters, but that's would be your minimum number of liters you would feed, just to not grow the animal, just to keep it alive. You know, obviously I would disagree with that approach. I would feed a lot more if possible. So, but I would be very conscious of what we're heading into and what you've already experienced, but also the temperature of -35, to feed a lot more, even during the first 2 to 7 days of life. Yeah. So I know a lot of people are having success with even with 2 time a day feeding, of even feeding 4 liters per meal. But it's okay if they don't consume it as well. Just give them some time to acclimatize to that diet. And that could happen where they skip the meal. That's okay. They're still young. They're still learning. So, but I would offer that milk early in life if possible.
Thank you, Mike. There was a couple of questions that came in for you, Greg, regarding the AMS. What's the maximum meal size, and can too big a meal size cause acidosis?
Yeah, these are great questions. And so, as I was mentioning last time, if we assume 500 grams a minute as our maximum meal consumption, and average box times of about 7 minutes per milking, if we assume 100% of that milking activity is also eating activity, we could calculate those cows probably could consume up to 3.5 kilos per visit. Now, again, that's with an average cow. So if the milking time is 10 minutes, that's different, but they probably do not eat at 500 grams per minute for 10 minutes. And in fact, the original research evaluating eating rate showed a sharp decline in eating rate as the meal advanced in minutes. So I think 500 grams is an aggressive amount, but if we look at maximum meal size, it's probably 2.5 to 3.5 kilos. We originally thought that high AMS allocations could increase risk for rumen acidosis, and we have rumen pH measurements in several of our studies, and we can never pick up a AMS visit event. What we do pick up is the PMR eating events, and I think this really highlights an important concept with AMS feeding, is that the bulk of the nutrients and the bulk of the fermentable nutrient supply comes from the PMR, not the AMS. And this really suggests that the AMS is a milk harvesting strategy rather than feeding strategy.
Thank you, Greg. So we have one last question for a quick answer from Mike. Mixing third and fourth milkings from fresh cows with milk replacer to feed calves. If this is pasteurized, does it hurt quality?
Yeah, I guess I have a couple comments about this question. The first is that, yeah, I know some other farmers feeding transition milk for the entire pre-weaning period, and actually they've had some good success with that. The concern here is that from day to day, if you don't have a lot of large numbers, you could be varying the macronutrient composition every single day. So I'd be very cautious of that and keep your eye on the total solids. And this comes into probably why this question is being asked, is that a lot of people are adding milk replacer or a milk extender is what the new terminology is, to balance out those total solids and maybe increase them a little bit. And it has been shown in the literature, if done properly, can increase growth because you're increasing the total solids of the meal. You're still under these thresholds and you're providing more nutrient density per volume of what you're feeding. However, the problem that comes up is when you're mixing a lot of these ingredients, you could run into some issues in abomasal health. This is all anecdotal. I just know that every farmer that has called the struggle, that has been struggling with abomasal bloat or health, has been mixing milk replacer and whole milk. And if it's not done consistently and in a clean fashion, it can cause a lot of problems. So that's my only comment to that, yeah, if you manage it properly and you're checking it every single day for total solids, I think it could potentially work. But if you're not, most likely you're going to run into problems through just delivering inconsistent macronutrients every single day.
Thank you very much, Mike. So we're going to wrap it up here. So another thank you again to our speakers. You were all great today. And thank you very much to our participants for joining in our first webinar. You've helped make it a success, and we're happy that there was such high interest. If there's any questions that we didn't get today, please feel free to submit your questions on the DRECA webpage, which again is in the public-facing Alberta website, for industry, DRECA, and then ask an expert. And we'll make sure that your questions get to the right expert and get answered. So thank you again for joining us today, and we hope you have a great rest of your day.